Good afternoon. Thank you all for being here. My name is Luke Ashley. I'm a rising junior at UNC Chapel Hill studying biochemistry. And for the past 10 weeks, I've been a part of the REU program working in the Lowry lab. So this summer, I've been investigating expression of NELFA and TAC3, which are genes associated with Wolf-Hirschhorn Syndrome, in developing Xenophus laevis embryos. One second. Here we go. So WHS, or Wolf-Hirschhorn Syndrome, is a rare disorder that affects 1 out of 50,000 individuals. Unfortunately, most die early on in development, in childhood. However, some do live on and into adulthood. It is a neurodevelopmental disorder characterized by craniofacial abnormalities, intellectual disabilities, heart and skeletal defects, as well as urogenital defects and seizures. Those affected tend to have what we call a Greek warrior helmet appearance, as you can see in these patients here. It is characterized by a wide nasal bridge, widely spaced eyes, a high forehead, drastic eyebrow arches, and an undersized jaw. Patients also exhibit microcephaly, which is an undersized head due to less brain development. It's important to note that patients tend to have phenotypes that vary in severity, as each patient will have his or her own unique set of WHS characteristics. So the WHS phenotype is attributed to irregularities on the short arm of chromosome 4. These irregularities can range from deletions to microduplications of the WHS genes in the WHS critical regions and flanking genes telomeric to the critical regions. So the 5 proposed implicated genes are WHSC1, WHSC2, also known as NELFA, LEDM1, FGFR3, and TAC3. It's important to note that not all patients have the same deletion patterns, meaning that some patients may have 1 or 2 of the genes deleted, some patients may have 1 or both of the critical regions deleted, whereas some other patients may have all 5 of the genes deleted. So if you were here earlier, you heard Rachel speak about WHSC1 and LEDM1. I will be focusing on WHSC2, or NELFA, and TAC3. NELFA, or negative elongation factor A, is a gene that has been identified to be deleted in most WHS cases, and TAC3, a gene that encodes for transforming acidic coil containing protein 3, is a gene that is deleted in many WHS cases and has been identified as a possible contributor to the disorder. So due to the protein products we see from these 2 genes, we have a hypothesis that deletions of these genes lead to defects in neural crest cell migration. So for that to make sense, we need to review what neural crest cells are. So neural crest cells originate at the roof of the developing neural tube, as you see here, and they undergo something called epithelial to mesenchymal transitions, which allow them to migrate long distances to form craniofacial cartilage and bone. You can see craniofacial cartilage and bone, skeletal structures, smooth muscles such as the heart, and other tissues. Yes, they migrate. So the main questions we're looking at for this project are, where are NELFA and TAC3 expressed naturally in the developing Xenophus laevis embryo? And how are craniofacial morphology affected by the knockdown or gene suppression of NELFA and TAC3? So our model organism is Xenophus laevis. It's a great model organism because it's relatively inexpensive to maintain. We get a lot of viable offspring on a regular basis, and the genes within Xenophus laevis are relatively easy to manipulate. So on the first day of an experiment, we inject the frogs with a reproductive hormone that induces egg production, and then on the second day we collect and fertilize eggs. Also on the second day we inject them with morpholino at the 2 to 4 cell stage. So morpholinos are a chemically produced compound that when introduced, they bind to our gene of interest. So they're antisense to the gene we want to knock down. They bind to the mRNA of our gene of interest, and then they basically render that mRNA useless when functioning in proteins. So no functional proteins are formed. So in essence, morpholinos will knock down or suppress the expression of our genes. So the first thing we want to look at and we want to know are where NALFA and TAG3 express naturally in the developing embryo. So to do this, we use non-injected embryos. So we did not inject them with morpholinos to knock down any gene expression. We want to see what's happening naturally. So we use whole mount in situ hybridization using an antisense DIG or digoxygen labeled probe. So in some of the final steps of the in situ hybridization experiment, we introduce a probe which is antisense to our gene of interest, so it binds to the mRNA. It is tagged with a DIG molecule that when we introduce an antibody, the antibody binds to the DIG molecule, and then later we introduce BM purple, which uses the antibody as a substrate to stain the probe. So essentially wherever we see staining wherever we see staining is where we see expression of our gene. So we use a probe specific to X-twist, which is a known neural crest cell marker. So here you can see that X-twist is expressed in the pharyngeal arches and in the developing neural tube. So what that's telling us is neural crest cells are migrating to the pharyngeal arches and the developing neural tube. So if our hypothesis is correct, then NALFA and TAG3 will also be expressed in these locations as well. So when we did an in situ using a probe specific to NALFA, that's exactly what we saw. So in stage 20, panel B, you can see NALFA expressed in the developing neural tube. You can also see that in stage 25. In stage 25, you can see NALFA expressed in the pharyngeal arches right here. And in the later stages, panels D, F, and H, you can see NALFA expressed in the craniofacial region in the developing brain. You can also see NALFA expressed in the developing gut right here. And so next we did an in situ with a probe specific to TAG3, and we saw similar results. So in stage 16, we saw staining of the developing neural tube, as you can see that here in stage 26 as well. And in stage 30, we saw diffuse staining of the pharyngeal arches. And in the later stages, we saw staining of the craniofacial region and in the developing brain. So now that we know where TAG3 and NALFA are expressed naturally in the embryo, we can try to use morpholinos to knock down this gene expression and then see how craniofacial morphology is affected. So when we did this, we looked at 5 parameters of craniofacial morphology of the genes that were not, of the embryos that had knockdown genes. So we looked at midface area, the area around the eyes and around the mouth, snout length, the length from the edge of the eye to the top of the cement gland, midface angle, the angle from the center of one eye to the top of the mouth to the center of the other, facial width from the center of one eye to the center of the other, and facial height from the top of the cement gland to the top of the eyes. So when we looked at these parameters, we saw that NALFA knockdown had a significant decrease in facial width, whereas some TAG3 preliminary data, knockdown data, shows that there may be a trend of decreased facial width for TAG3 knockdown. So looking at some embryos here, we see that compared to the control, alpha knockdown and TAC3 knockdown had shorter facial widths. And we also see that alpha knockdown had a significantly smaller mid-face area than the controls. And our TAC3 preliminary data are showing that there may be a strong decrease trend in mid-face area. So taking a look at some of these embryos, we see that compared to the control, alpha knockdown and TAC3 knockdown had a smaller mid-face area. Further, we see that alpha knockdown shows a significant decrease in mid-face area on mid-face angle, whereas our TAC3 knockdown preliminary data did not show any change. So looking at the alpha knockdowns here, we see that compared to the control, alpha knockdown had a much smaller mid-face angle. And lastly, we see that alpha knockdown and TAC3 knockdown showed no change in facial height and also showed no change in snout length. So to summarize, we see that alpha is expressed in the pharyngeal arches, the neural tube, and the craniofacial region. And we also saw that alpha knockdown embryos show significant decrease in facial width, mid-face area, and mid-face angle. We also saw that TAC3 is expressed in the neural tube, craniofacial region, developing brain, and diffusely in the pharyngeal arches. And we have some TAC3 knockdown preliminary data that shows a possible trend of decreased facial width and mid-face area. So this data is important because as we see how these genes may be implicated in neural crest cell migration, we can possibly use this data to further our studies to maybe later in development of these studies to find treatments for people with WHS. So future direction, we're going to finish the TAC3 craniofacial analysis to see if these trends that we saw held up. We're going to focus on the last gene of the 5, FGFR3, so we'll do the same experiments we did with the other genes, the time course in situ hybridization and craniofacial analysis. And also we'll do some cartilage staining to see the effects of knockdown of each of the genes in cartilage. So I'd like to thank all the members of the Lowry Lab, specifically Rachel, Ali, Fred, Megan, and Sangmook. You guys have been an amazing help. And of course, Laura Ann for accepting me into her lab. And Ken Birch, who co-produced this program. And in terms of funding, I'd like to thank the March of Dimes, American Cancer Society, and NIH, and the NSF for funding me this summer. And with that, I'd like to take any questions.
Questions? Yeah, so when you first introduced the genes that were implicated, you had a visual, like the third slide, where you have like the strings of nucleic acids. Yeah.
Yes. Yeah.
So is that just showing where they are on the chromosome?
Yes. So alpha is first, or WH is C2, and then so this is the telomeric end, closest to the end of the chromosome.
Okay.
Do these frogs live into adulthood, or do they die as tadpoles?
So we basically culture them and remove their heads at certain. We fix them, so we kill them, and we remove their heads at certain stages in development. So if you see here, so with these pictures, this one was fixed at the blastula stage in stage 20, so just different stages in development so we can see where the genes are being expressed.
How do you fix it at that stage? How do you basically,
Kill them?
yeah, how do you
So we put them in something called paraformaldehyde that will preserve them, so it kills them and preserves them.
I'm sort of confused about the bit about knockdown. How do you actually know the craniofacial differences you saw due to knockdown? I didn't follow that bit.
Oh, okay, so we use validated morpholinos. I don't have the validation here, but the morpholinos that we use are validated. And so once we know that the morpholinos are validated, we inject them into the embryos to essentially knock down the gene expressions. So the way we know that our craniofacial data we saw is due to knockdown is that we did rescue experiments. So how rescue experiments work is we simultaneously inject morpholinos and mRNA of our gene of interest. So the way it works is that the morpholino will knock down the gene expression and the added mRNA will supposedly reverse the effects of the morpholino. So the mRNA we inject will be translated into protein, and the mRNA that was naturally in the embryo will be rendered useless. So it's like a balancing act. So we did that with varying concentrations of mRNA, and then we looked at the craniofacial morphology of these rescue embryos and if they had no significant differences from the controls, then we essentially rescued the phenotype, and we know that the knockdown came from the morpholinos.
Okay, and the mRNA is what does the rescuing?
Yes.
Okay, that makes sense. Any other questions?